Mary, I might need to take that one to Robert T. But let's take it to the Slack maybe if we can't find a solution. So I'm just seeing it's in NF. Oh, no? What? Okay, so this Conduit Tools is bioconduit. Seriously? This? Where's this coming from? Okay, so if that's from there, then... So I'm just trying to figure out how Robert's been doing this. Oh! So it's got the same dependencies as the main Snippy package. So I'm just wondering. Okay, I'm going to have to take this to a higher level of expertise because I'm not getting it. The bush kangaroo, okay? So what is Snippy? So many years ago, there was a TV program called Skippy the Bush Kangaroo. Okay? It was an Australian TV program, okay, about a boy with his friendly kangaroo, okay?
Is that in the 1900s?
Is that in the 1900s? Thank you, baby in the corner, okay? Yeah, we don't need to comment about age in this class, thank you very much. So, this is an Australian TV program. Then at some point in the, I don't know which years, this man was born. We're not going to talk about years. This is Professor Torsten Seaman, who wrote Snippy, okay? And he has a sense of humor, so it relates to SNPs, you know, single nucleotide polymorphisms, and he's Australian, so he called it Snippy like Skippy the kangaroo, okay? And what this thing is doing inside is that it's doing bacterial mapping and variant calling, okay? So, and it has never been published in any journal anywhere, but it is widely used. This is not, so mapping and variant calling, okay? So I'm sure that we've gone over this before, mapping and variant calling. Most of you have heard about like this. And for mapping, oh, there's a big fancy pen today, okay. For mapping, so obviously we know that before we do mapping, we do quality control. We did that with the clean your reads using FastP. And then the next step is typically variant mapping, for which there are tools like BBMap, Bowtie, and BWA, okay? BWA from Heng Lee. And BWA has also never been published in a journal anywhere. Okay, it's one of the most widely used pieces of bioinformatics software, and it has never been published. Okay, because Heng apparently was planning to publish it, got some reviewer comments, didn't have time to deal with them, and they never ended up publishing it. But it's still very, very widely used. From mapping, once you've mapped your reads, then you can do variant calling. Okay, inside of Snippy, it uses a tool called Freebase to do the variant calling from Eric Garrison. And then after that, if you use a GenBank format input file, Snippy uses SnipEFF to compute the impact of the variance. Okay? So this is the tool, the command line tool, and if you look at it describes how to run it on the command line, but the bit that I'm interested in is this list of what outputs does it produce, okay? So Snippy produces a whole lot of outputs, including VCF file, a BAM file, which is your BAM is your aligned bacterial reads, okay? And consensus, okay? So it produces multiple consensus files. So the consensus but also a consensus only has the substitutions. In other words, only the single nucleotide substitutions, not the insertions and deletions, okay? Which is useful for when you want to do phylogeny, because then you can just use it without having to realign your multiple sequence alignment, okay? Why is this useful? Because when we go look in the outputs of, let me just save this. Let me just go, I think this was my Burberry folder, yeah. If we now look at the Bactopia Snippy folder that is created after running this workflow, then we again see the different files for each sample, okay? And if we look inside those files, so like this, it's blah, 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 okay? Then we see no longer, before we saw a main folder inside the sample directory, because we were running a part of the main Bactopia workflow. Now we see tools, because this is add-on tools. The Snippy is not the main Bactopia workflow. And tools, and then inside there's Snippy, and then we see this. Okay, so you'll recognize this name NC016445. That is what I said when I said run Snippy. Then I said that the run name must be this thing, okay? And the reason for that is because the Snippy, you can obviously use Snippy to align against different reference sequences, okay? So if you don't set run name, it will just call that folder Snippy. So then you'll get a Snippy/Snippy folder. But I like giving it a meaningful name so that I can remember which reference did I use for this run, okay? And then inside that folder, then you can see all of these files, okay? And these files, broadly speaking, correspond with the files that are mentioned on the Snippy webpage, okay? And so we see here consensus.subs, the raw and filtered VCF, then they compress the VCF, and so on and so forth. BAM and the BAM.bai, which is the index of the BAM, and yeah, the different like FASTA files aligned on consensus and subs and things like that. Okay, so we see them here, and so we can see from this what the utility of these different files is. Okay, so over here, this is just a tabular variance. This is the variance in bed format and so on. They've all been compressed. Okay, and these are produced for every individual sample. So we've got one of these for every sample. Okay. Then the other thing that we get. Okay. So this is what you're getting for each sample, and you can look at the VCF, and we will look at the VCF later on. But the other thing that we get is a Bactopia runs folder. Okay. Now, if we go back through what happened with the Bactopia run, then we see that for each sample, it ran Bactopia. Okay. In other words, it ran Snippy inside Bactopia. But then it ran Snippy core. Okay, if you read the Snippy website, you'll find out that what Snippy core is doing is that it reads the outputs of individual Snippy runs and it finds the core SNPs. Okay, so what the core SNPs are is SNPs which are present in all of the samples. Okay, that there's some information. So in other words, according to Torsten's description about Snippy core, if you call SNPs for multiple isolates from the same reference, you can produce an alignment of core SNPs which can be used to build a high resolution phylogeny. And the core site is a genomic position that's present in all the samples. Okay, what it means by this is that there's not any sample in the collection where there's maybe not reads at that position. Okay, it doesn't mean that there is the same variant at every position, but it means at least you've got information as to whether the variant is present or absent. Okay, and it just uses variant sites, and that's the core SNPs, and this is used to produce the variants that feed into the phylogeny. Okay, we see when the Snippy process runs, then we see down here at the bottom IQ tree, which you might recognize from other bioinformatics courses, is a very commonly used phylogenetics, maximum likelihood phylogenetics program. Okay, but there is something in between called governance. Okay, and snip this, but we'll get to that just now. Okay, so what governs is doing is that, remember, bacteria are recombining their genomes. Okay, they're exchanging bits of genome, and recombination is a thing in bacteria. We expect that this Vibrio is clonal, but recombination is always a possibility. Okay, and what recombination does is that you have a recombination breakpoint. Imagine the genome now being made up out of different pieces, then reads that align around the recombination breakpoint are going to have lots of variation, because one side of the read, because if we compare that, if we think that like the reference sequence, okay, it was all looking approximately like one thing, but now this sample, there's been some recombination, right? Then the reads are going to map to somewhere in the reference, but it look like there's a lot of variance showing up here, okay? But that's not true variation from a, phylogenetically useful point of view. So what governs does is identify likely recombination breakpoints and filter out variance around those positions. Okay, so governs is a recombination filter, and it's very commonly used, very common procedure to do your variant calling, create your core phylogeny, and then pass it to governs for filtering. Okay, before doing your phylogeny, because what this would look like if it wasn't filtered out in your phylogenetic tree, then you'd have a long branch, because it looks like mutations are happening when what's happening is that these are not mutations being introduced by biological processes, but they are false mutations from incorrect bioinformatics, effectively. Okay, so you get these like kind of long branches. So gubbins is being used to do that filtering. Snip disks, if you've worked with some of our previous workshops, you'll have seen snip disks. It just computes a distance matrix of snips between all the different samples. Okay, it's also one of Torsten's tools. And then finally, dump software versions that gets run every time because basically you want to keep track of the software versions. Okay, so if you see here, each individual Snippy run was running for one sample, but everything from Snippy core onwards is running for all of the samples. So that is what ends up. So we saw yesterday in our Bactopia runs output, it was just the Bactopia, it was just the Nextflow log that let us see what processes Nextflow had run. Okay, but now if we look in the Bactopia runs directory, again, we'll get one of these files for every time that we've run this workflow. So if I run it another time, I'll get another file like this, okay? But so you must pay attention to the date and time of the run. I actually don't know how to, I wish there was a way to just choose the name for this directory. Maybe there is, I just don't know it, okay? But this is the combined output directory. If we look inside of it, then we see the steps that I mentioned that get run for all of the samples. So there's the Nextflow reports, but there's gubbins, Snippy core, snip dist, and IQ tree. And so that's where all of our outputs from those tools are ending up, okay? And so in IQ tree, we have a tree file, an IQ tree file, and all of these things which are generated from the core phylogeny, okay? So if we look at the IQ tree file, this is the actual log of IQ tree, okay? And if you've run IQ tree in Galaxy, this will be familiar to you because it's just a visual. It's an explanation about like how IQ tree is running, and one of the things that's kind of nice, it tells us which model it shows. It shows HKY plus F, various parameters, and then there's a simple text version of the tree. Okay, so we can just have a quick look here. The reference is off on its side. This is the reference was chosen as the out group, okay? So if you recall from your phylogenetics lessons, everybody here knows the basics of phylogenetics, right? Is there anybody here who's never been taught phylogenetics? No, okay, yes, okay. So an out group in phylogenetics is you choose and what you expect to be an ancestral sequence. So that if you don't choose that, then you don't know like which came first, you know, in terms of, because all we can talk about is difference. Then we have an unrooted tree. It says this tree is unrooted, but basically what you do is you use this to kind of give a direction to your tree. So if you say that reference, in our case, remember that the reference is a sample collected early on in the history of cholera in Haiti, then we know that every sample, all of the other samples are more evolved. The evolution has a distance. But if we didn't know, if we had like a bunch of different samples, remember that maximum likelihood methods, all that they're working from is the genetic code. They don't have any idea of time, okay? So if you just gave a bunch of different samples to a maximum likelihood method, you didn't give an outgroup, it wouldn't know which one comes first, you know. This is not a time tree. This is a tree in terms of genetic distances, okay? And we can also see over here in the numbers, bootstrap values, okay? Anybody remember what a bootstrap value in a phylogeny is? What is a bootstrap value? What do these numbers tell us? The number of times that this particular split in the tree has shown up when you recompute the tree. Okay? So that means that this branch is quite certain, but whether this is the correct split, not very certain. Okay? So I don't know why there's two numbers here, but anyway. So yeah, it's running IQ tree with its bootstrapping thing. And then in the same folder,
It explains it in the text.
no, is it?
Numbers in
Okay,
parentheses.
so I don't know what SHALRT is, but ultra-fast bootstrap is the second number. It says it's unrooted, but it's...
There are two.
Oh, is there?
I think those are the concepts of the two trees.
Is there? Is the rooted tree below that one?
Yes.
Okay. Okay. Okay, so this is the rooted one. Yes. No? Yeah, the consensus tree. Yes. You're right. So this is the original unrooted tree, but this is the consensus tree after you've run 1,000 bootstrap trees. And it says here. So this is its consensus, and this is its final bootstrap likelihoods of these splits. Okay? At these positions. Okay. And what we can see here is just a grouping, right? Because we haven't included time into the equation, but we can see a grouping and the emergence of clades. Okay? So, and remember the thing with phylogenetics is that phylogenetics is about ruling out, not ruling in. Because there's always missing data. There's always a missing in-between data. So what we can say is that if we take this sample over here, okay, and then we have somebody who comes with a sample more similar to that, then we can say that no, there is not transmission happening. This is not a transmission from here to here, okay, because they're two different clades. But what we can't say, this one and this one, we cannot say that this one transmitted to that one, because there could be missing pieces of the puzzle that we don't have. Okay? This is, of course, a very small phylogenetic tree, just because I didn't want to take up the whole day running Snippy and things like that. Okay?
The numbers in brackets.
Number in brackets is your bootstrap support.
Okay. And then when you look at the one that says 100% and the one that says 40.
Yeah. So what that means that this split is not showing, this particular organization of the tree is not showing up all the time. So which means that I think that what's not showing us is what the other options here are. So maybe, I don't know if it's that the order of these three can be in different positions. So that maybe sometimes this one is here, you know, or something like that. So remember that, like, even if you've got, remember that in maximum likelihood, even if you've got a polytomy, in other words, where it looks like the tree should branch like three ways, right? Because there's not really much evidence for this one versus that one. The system has to choose a sequence of two-way bifurcations. So we'll see when we look at those things in the SNP distance matrix what we can say about the number of SNPs between these samples. Okay?
It could also mean that the one with the 40, that you have a lot of missing sampling points, because as you can see, we only have 5 samples, is it?
Yeah.
1, 2, 3, 4, 5.
Five.
It's very clear that this is not part of this going all the way back, but then we can't place this one. But it's more likely to be related with this, but then we have a lot of missing data.
Yeah. Everything in between here is missing.
Yeah. So we don't know what you could ask EPI people, for example, a public health officer, is that did you collect samples from all locations?
So the relatedness now comes to those ones up or to the reference?
So the reference.
It's based on the reference. Right?
So
Not to
so
the
what we've got here is that the reference is clearly in its own claim. So we don't.
It's only been used to guide
So
the tree.
it's related to the other one.
Yes,
You
the reference
might be
here.
asking what 40% actually represents.
It means that, but remember, 40% is when you calculate the tree, 40% of the trees had that particular
The agreements.
agreement. Okay, but the reference. So what does it mean that the reference, okay, is over there and that branch length is that? Okay, because in terms of time, we know that the reference was collected before any of these guys. Okay, but when we are computing, and George maybe guide me if I say something wrong here, but when we're computing that phylogeny, is that we don't know which SNPs the ancestor had. Okay, so it's what it's putting here with the absence of time. Okay, so maybe what should be the case is that reference is all the way to the left because it's earlier, and that this branch is longer. You know, because actually we know that the evolution. So what it doesn't, because it doesn't have the sense of time, it's putting another alternative hypothesis in front of us, which is that actually the reference accumulated a whole lot of SNPs. That's why you've got this like relative to the inferred ancestor. But we don't have the inferred ancestor, so that's why it's not getting, it's drawing it like this. If you were to now scale this tree by time, the shape of the shape of the tree might stay the same, but the branch lengths might be shifted around.
So the branch lengths here are simply genetic distances.
Yeah.
These days. And the length, so one thing we're missing here is a scale that tells us the relative difference between the ratio of the length to the number of mutations that have been accumulated over time.
It doesn't show us down here, no,
Does it give us a
doesn't give us a scale.
a scale? So it's a bit difficult to interpret without a scale, like how many mutations have accumulated over time. But as Peter is saying, in this case, the topology here doesn't matter. I wouldn't care about where the reference is. It's a computational process. You have to place a reference somewhere. It's completely meaningless, but it does show that indeed there are loads of differences between these samples and the reference.
Yeah.
Okay. There's no relatedness. So all these did not necessarily come from the same epidemic. Where there's very little ancestry, which also could mean that there's a lot of missing samples.
Yeah.
Okay, that's one way to interpret it.
And just to give you a bit of actual background here, the reference is about 2010, okay? The samples here, the earliest is, I think, 2014.
And locations? Where is that
And
reference from?
so they're all from Haiti,
Okay.
but the time difference between everything that is an actual sample here, except for reference, is, I think, 1 or 2 years. But the time back to the reference is 4 or 5 years. So, but of course, the phylogenetic program doesn't know that. Yes.
If you change the reference,
To what?
To what?
like you can look for another reference and use it, probably it could provide more information.
But I don't think changing one sequence is going to be the trick here. The trick is make it more dense. Fill in the gaps. So
So if you change the reference to an older reference, do you think the topology of this tree will change?
Yeah, it's a good question. If we took the 1975 Bangladesh one
Yeah.
that is often used as the
reference.
the reference for Vibrio cholerae.
Do you think the topology will change? Why?
I don't know. I think it will change because the other one is much
No, the
older
topology.
than
But
this
the,
one.
but the,
So
the lengths can change, but will the shape change?
the shape might not change.
It will not change. Okay? Because the relatedness here between these samples, they're still maintained because they are from the same outbreak. And the only reason we're using the reference is for us to be able to align these sequences against something. So that's why it's in there.
Yeah, maybe this is reference-based assembly, right?
Yeah. So if we calibrate this time, this tree, maybe we'll do that, I'm sure.
With
And
the
put it
Backdate or something. We
Yeah.
might do that.
back there. And reroute this. What you see is that the if you take a longer an earlier reference, the distances will be very long. Okay, what you call them, the branches to the reference might be.
I should actually grab the something else and do it, but yeah.
If you took one of these as a reference, for example, and you argue this is my reference, what do you think will happen? If you took, for example, you could take this is the most different one. I could redraw this and use this as a reference if I
If we assemble a genome out of that, yeah.
if I assemble that genome and I could use this. It's the most sort of far back in this context. But also you have to remember in how you're interpreting it is you have to have other data, other metadata. So my quick question, would just be that is this from the same location? Is that from the same location? From a public health perspective. But not to distract Peter.
No, but this is exactly. So the thing is this, that this is a very toy tree, and you make it much denser, right?
Yeah.
And when you do a denser tree, then you can start seeing like stories of, because what happens with outbreaks is that you have interrupted expansion. Okay, so because the chance of one person, the chance of infection is distributed. So some people don't infect anybody. Okay, some people infect many, and you've got this distribution. So what happens here is that so somebody with cholera is catering a wedding, sudden expansion, and that particular clone that is in this caterer suddenly has many replicas. Okay, and now, or sampling. We have a rural area and an urban area. Okay, in the rural areas, most people are not getting healthcare. Perhaps they're passing away. No sampling is happening. In the urban health facility, we are sampling a lot. Okay, so we get this density of sampling in the urban areas, and so this is where things get tricky, because now if you get this dense sampling, and then you've got a few other things, we might not know the true shape of the evolution. I mean, the example I always give is so when the variants alpha and beta identified in December of 2020, okay, then it became a thing. Everybody wanted a variant, like Oprah, like you have a variant, you have a variant, you have a variant, okay. So a California lab announced that they'd found a variant of interest, okay. Now remember, the characteristic of the variant of interest was that it was doing a sweep and replacing. Previously, where you had so much genetic diversity, you were finding that all of those other lineages were disappearing from your surveillance, and one new set of lineages was taking over. So, and then they made this announcement that they found like 80% of their samples in this California lab was this one variant, okay? But then a little bit of explanation turned out that they were prioritizing genomic surveillance of vulnerable populations, like, for instance, old age care homes. So they collected the majority of their samples from one outbreak within a old age care home. So then it looked like, because they had a big outbreak in this care home, but you were having outbreaks elsewhere, but they just weren't being sampled. So it looked like there was this variant that was expanding, but it was just a sampling bias issue, you know. Whereas the original hypothesis for the so-called alpha variant discovered in the UK, at that point of the pandemic, the general rule that we were all working with was that the genetics of the virus didn't make much difference at that point. It seemed to all be the same from a epi point of view. So the first hypothesis that the UK people had was that it wasn't a variant issue, it was a behavior issue. That maybe, despite the lockdown rules, people had come together for a big gathering, okay? In fact, in the headline of the article in The Guardian about it was that, did they have a big rave in Kent or something? Because Kent in the UK is where they first found the variant. So they thought it was people coming together and that causing the expansion, not because of anything about the virus, but because of the way people behaved, okay? And then later examination of the other contextual data from the field investigators showed that no, there was no unusual behavior that could explain the sudden increase in prevalence of this one lineage. And then much later, took a lot of other work, they found that there were virological characteristics of the virus that allowed it to expand. And if I recall correctly, one of the features was that it was able to evade the immunity from prior infection better than other variants, you know? But this investigation took, like, putting all of the different skills together, the epidemiologists and the lab people, bioinformaticians and so on, which is what one needs to do. And also why you need these integrated data systems. Two questions. Yes. And the first question is?
Okay, the first question is, as George was saying, if we could reference that sample, 370, are you able to see the 295?
295.
253, I mean.
253, yes.
That one you can see.
This one, yes?
Yes, the one up.
So if we were to do what with it? So we've got 2583 over there, then
So
it's
you
373,
basically see the two. Like, we use 370 as a reference gene.
370, yes. Yeah.
Are we able to pick up the two samples?
So look, I still think it's likely, given that we'll see from the slips just now, but I still think it's likely that we'll see 253 and 373 grouping together,
Yes.
and 260 and 364 grouping together, okay?
Yes.
But you raise a good question, which is that if you choose something in the middle of a tree and you make that the outgroup, okay, then imagine that you have a sample, okay, and this sample has variants that are in common with many of your other samples, okay, then it will confuse the program in terms of like how to organize the tree, because now it won't know, is this going this way or that way. That's why we choose an outgroup, we choose something that is a bit distant. So when we're doing Mycobacterium tuberculosis, we use Mycobacterium cannetti as an outgroup, because it's from like somewhere away, you know. If you were trying to root the phylogeny of the great apes, you might choose a monkey or something like that as an outgroup, or something like that, you know. So yeah, just in order to, but you don't. But on the other hand, if you choose something like, I remember early in the pandemic building some phylogenetic trees where we included bat viruses. Okay, so if you take some of the bat viruses, okay, or maybe the virus collected from pangolins in China, then all the branches collapse of the human ones, because the human stuff was so similar to each other that you just can't see what's going on, because the other sarbecoviruses are like quite, like if you look at like the bat viruses, the closest bat virus is, I think it's about like 800 to 1,000 SNPs distant from SARS-CoV-2. Whereas if you look like SARS-CoV-2 to another SARS-CoV-2, especially early on in the pandemic, it was just 2, 3, 4, 5, 6, 7, 8, 9, 10 differences, So you must choose something that you don't want to collapse your, depending on what your question is, right? Here we're investigating an outbreak in Haiti, so you choose appropriate for Haiti, right?
Just on that topic, just to
Yeah.
for those who are working on SARS-CoV-2 data, Zhang Zheng has released a treasure trove of genomic data from China very early in the pandemic. And I'm sure there will be now papers trying to reroute and reanalyze data from the early pandemic. So we have those.
But it's not that early. It's the 25th of January.
Yeah, but
It's early compared to some things, but it doesn't change the route because it's from
yeah,
end
don't
January.
change the route. But if you have one of those early samples and you want to reanalyze those data, at the moment we only have like SARS-CoV-A, and then we go to the B's.
Yeah.
We have very little knowledge about what happened between the A and the B. The G604 mutation prior to that.
Yeah. So there's still some debates as to whether there is anything in between. But yes, I mean, but I also like this stuff, and then but yeah, old samples are useful for other things. But anyway, phylogenetics it's this whole other thing, and yeah.
You need an entire course on that.
But the other thing about phylogenetics is what can really mess up your phylogenetics if your alignment is wrong. Okay, so if your alignment is wrong in various ways. So, for instance, if I had, and I always have to come back to this, if I had a poor quality sample that was missing a third of the genome because I didn't get enough sequencing reads to cover it, where would it approximately fall on the tree? To the right or to the left? To the left, eh? Because it would look like it's more basal, okay? If I have too few mutations, right? That's where this long branch problem comes in. If I don't analyze correctly, so if I include poor quality data and I have many apparent mutations, then it will shift me to the right. I will get a long branch, So, Tracy had that example where there were some organisms that were not Vibrio cholerae in the data, okay? And if you read the preprint related to those samples, okay, then what they tried to do was map those samples against the Vibrio cholerae reference genome, and they couldn't, the reads didn't map, okay? And so they excluded them from further analysis. But if it was close enough that the reads did map, okay, then you'd actually see it in the phylogeny. So I had the one time I was reanalyzing the Mycobacterium tuberculosis data, okay, from Indonesia. I built the phylogeny and I couldn't understand that I had these massively long branches, okay, just 2 or 3 of them. And I went back to the data, and so typically in one Mycobacterium tuberculosis sample compared to the A37RV reference, you see about 2,000, 3,000, 4,000, let's call it around there, depending on which lineage you're dealing with, single nucleotide variants compared to the A37RV reference sample, okay? And I went back to look at my data, and I had 30,000 variants in these branches compared to the other branches. And I went to look closer, and I found that actually very few of the reads were mapping. The map depth was very, very low. And further analysis showed that what I'd almost certainly picked up in the data was a non-tubercular mycobacterium. that they'd included in their Mycobacterium tuberculosis data set. So it was close enough that it mapped to the Mycobacterium tuberculosis genome, but it didn't map well. So that looks like a whole lot of SNPs because it's actually like another species, you know. And I went back to the people who generated the data, and they assured me that they'd done all the biochemical tests and whatever to make sure that this was Mycobacterium tuberculosis. But we've never resolved this question, how that ended up in the data. Because I mean, I don't know how the discussion went on Tuesday, but how did this Vibrio cholerae end up in a Vibrio cholerae sequencing run? You know, I mean, to me, it's a gram negative rod, so maybe under the microscope it kind of looked the same, you know. But I don't know. I don't know that the lab process is well enough. But so if contamination and all of those kind of things can then extend branches with incorrect information, okay. So anyway, so this is your phylogenetic tree. But let's also see what differences do we have. So. Oh, there was a second question. Sorry.
Sorry for taking your time.
No. I have a. You know what? Go ahead. You're not by far. I have a colleague. I won't say from where. Doesn't ask questions on that Slack that you guys all join. She doesn't ask questions in the general discussion where anybody can answer. She just sends me individually questions, and I'm like, ask in the channel, please. Make it a community. But next time, I'll open my Slack and there's another question. But anyway, go ahead.
Okay. So, looking at the percentages of
Bootstrapping, yeah.
can I relate the number of mutations based on the percentages? Let me say, if I have two samples, let me compare sample 253 and 370.
Mm-hmm.
So it means that 370 relates to that just 40% of it.
No. This is the length, but as George said, we don't have the scale bar. The length gives you how many mutations we're dealing with, but not the bootstrap values. Okay? So if we take this, okay, so now we can take this is the IQ tree file. I want to see if this works if I do it with MicroReact. Probably not, but let's just see. Let me see if I've got a mouse. Where's my mouse going? Okay. So if we look at the tree file, this is a NUIT format tree, okay? But even here, it's not giving us in terms of the number of mutations. It's giving the branch lengths, okay? According to some scale where this numbers must relate to the number of mutations somehow, but I'm not quite sure how, okay? But that's why I wanted to switch to RStudio, okay?
So can I say the one with the lesser number might have more mutations?
No, you can't say anything for Bootstrap. You can just say for the branch length. Branch length equals mutations, not Bootstrap. Bootstrap is how certain, given the data, that the shape is right. Length is how many mutations we're dealing with.
Okay.
Yeah?
So I just wanted to ask that, so since we know that the low quality rates of samples is going to...
Sorry, yeah, a bit louder.
Okay, so... Sorry. Okay. So we know that samples with low quality rates will impact the phylogenetic.
Yes.
So does the SNP have a way to remove those adventure in
It
the sample?
does.
It does.
It has a built-in traumatic, but remember we ran clean your reads before we ran this.
Okay.
And that's what that was doing.
Okay, so when we run the clean your rates, so if we check, of course, we know the samples that it has. So when we run the SNP.
Oh, so what we haven't checked, what we didn't do, is look closely at the multi-QC output to decide to exclude certain. Yeah. So what we didn't do is a lot of pre-checking, right? So what we could have looked at is maybe like in the multi-QC output, you find some that the number of reads present is too low, or the QC, the GC value is off, or there's some other work that makes you think don't take this analysis further. And you guys who were on the CoalGen training, I've looked at some of the CoalGen data, and a lot of them I had to exclude from further analysis. What were the main issues that you found with that caused you to? Because you guys must have had the same thing. What did you do to decide which samples to take into later analysis?
So because for the back page on Terra,
Mhm.
you actually do the assembly first, generate a multi-QC, and then so we further steps down, we check the multi-QC result, and then filter the samples and speak, and then put those into the next process.
So Backpage Assemble, by the way, does something very similar to Snippy.
Yes.
It's pretty much the same
Yeah.
procedure. But then you looked at the multi-QC, which has got your BAM quality report output. And in fact, I was going to talk to Robert and say, can we include like the a multi-QC? Can we include because the multi-QC that they're running in Backpage is looking at BAM QC. It's looking at I think it also uses FastP for filtering. Was it? I can't remember what it uses. It uses something. Look, it uses one of the quality trimmers. I can't remember which one it is. It uses FastQC as well. What I was thinking is to add a few more of the QC steps into this pipeline. Okay, what's missing in all of these pipelines is using something like Kraken to try and identify what species you're looking at, or if you run the main Bactopia pipeline, which crashes my laptop because one of the steps takes too much RAM. Okay, it does a de novo assembly, but the thing that's very useful there is that it does what's called a sour mash sketch, where it basically tries to predict which species you're looking at based on comparison to a database called GTDB. And I found that that's very useful because it basically, if you do that and it doesn't show up as Vibrio cholerae, don't take the sample further. Okay.
Which workflow is that?
The main Bactopia assemble workflow. If you just run Bactopia without the -WF command, I'm hoping I'll have time to show it tomorrow because I can't run it on my laptop, but I run it on the bigger server. Okay, and then that does various sketching and MLST prediction and things like that. Yeah.
Does Gambit do the same?
Does what?
Gambit. In Terra, we use Gambit to predict the species.
I don't know. I don't know Gambit. Could be.
Yeah, it does the same.
Yeah.
Terra has multiple number of databases, so it takes Gambit, that mash and another one, just to tell the species.
So is it doing a mash? Because so the ones that I've seen are so obviously MLST calling, which you can do from whatever marker genes are present, and then you can mash sketch against RefSeq, and then the sour mash against GTTB. So this is what Bactopia has got. But yeah, so the general errors that I've seen is just too few reads to work with. Okay, a fair number of the CoalGen data sets had too few reads that I looked at, and then it's not Vibrio. And I mean the thing is, it's and that speaks to stuff that's happening in the lab, right?
Yeah.
That's happening upstream in the lab and improving lab processes. So I'm not going to go into which country was which, but like some labs, there were a lot of samples that didn't pass QC, you know. And it's not just with that. I had some people came and asked me about hospital outbreak related data, and I looked at their data and I was also like, look, a big chunk of this is just not possible to analyze. which means that your DNA extraction went wrong, maybe your culturing went wrong. You know, there's so on the other side of the bioinformatics, I think that there's a lot that needs to happen in terms of lab strengthening to get us good data to work with. Because, I mean, I'm using clean data here, but I've seen a lot of bad data. When by the time it ends up in the kind of published literature, it's all looking clean and beautiful and lovely and wonderful. But yeah, so one of the things that I think, so just for on the lab side of things, okay. So there's lots of different groups, especially ASLM has spoken about like lab strengthening, but I don't know if enough of that is really happening in the NGS labs, because there's so much lab strengthening to talk about, right? So there's lab strengthening for PCR, lab strengthening for bacteriology, lab strengthening in all sorts of other ways. But like, is there enough like training for people to like learn how to deal with the errors that they see on NGS? Because I've seen this a lot with Oxford Nanopore. They get the nanopore sequences, and I'm more familiar with the nanopores than the Illuminas. They get the nanopore sequences, and then the performance is not good. And I don't feel like, or at least in my experience, but I'm kind of outside of the lab space, there's not enough really support to help people understand how to get better performance out of the equipment. You know?
I've heard a lot of complaints in that direction.
So, I mean, George actually, so I remember, George, you know, you guys did the paper on optimizing the Arctic for
Low cost.
and for low cost, but besides that, I remember you guys early on in the pandemic working with, because I remember the early Arctic protocol, the annealing temperature was very sensitive, right? You have to get just the right temperature in your thermocyclers for it to amplify nicely, okay?
Yes.
Else you had like amplicon dropouts and things like that.
that.
So, and I mean, you guys did all of that work, but another lab that I visited in the East African region, they were just about ready to throw their nanopore away because the performance that they were getting was so poor, you know. And I was quite frustrated because I said, look, you know that within your region, there's actually people that know this machine very, very well. But like, is ASLM not providing any forums for people to talk to each other on the lab side? Is there no networks to go to if you say the sequence is not giving me good results? Is there nowhere to turn to?
No, we usually contact nanopore representatives within the region.
Yeah, I don't think that's very effective. They'll just tell you that like,
Yeah, they will tell you what you want to hear.
or what, yeah.
It doesn't.
No, I'm not. I must admit, I'm not terribly impressed by having spoken to company representatives, because they all basically say, "Oh, we're selling you magical machines."
Because my mission was always like, you have a sample, the depth looks okay, the coverage is 99.9%. You run the QC on it, and it will tell you it's bad. So you're like, what now?
Okay, but that if you've got good coverage and you've got good depth, then often it is like a different problem, and then you have to think about the bioinformatics and there's more complicated problems there. But what I'm often seeing is that I can't get good coverage, I can't get good depth. Why am I doing a sequencing run and I'm getting like bad, like I'm getting like a 50 megabyte file out of my sequencer, you know, or nothing. Those are the kind of things that I'm seeing from people, and both in that Nanopore and Illumina. And I think, like, I mean, the other things that I'm seeing is, why is 30% of my Mycobacterium tuberculosis sample human reads? I'm like, do you guys wear gloves in the lab? So it's those kind of, like, you cannot, like George told us last week, you cannot fix bioinformatics on its own. It's a process. It's a process thing, right? And I mean, like, when somebody sent me a sample of Mycobacterium tuberculosis that was 20% of a pathogen, no, not a pathogen, of a bacteria that's normally found in the human gut. I was like, guys, did you go to the toilet and then go to the lab and not clean or something? I mean, because what is this? Mycobacterium tuberculosis is a pure culture. You grow this thing from pure culture. If you contaminate with anything, it's going to show up in your sequencing. But yeah, so I don't know. I think that like there's a lot of gaps, and it's not just bioinformatics gaps. But anyway, let me get back to the data. Okay, so we've got this data. The other thing that I wanted to show you was let's not stay in one world, okay? I want to look at the SNP distance matrix, okay? So if I look at well, I've been looking at the IP3 results, and this afternoon I'm going to do more work with the IP3 results. But let's have a look at the SNP dist folder, okay? Sorry. And within the SNP dist folder, there's a file called core-snpdist.tsv, okay? And that tells you the actual number of SNP distances that there are between the different samples, okay? So we were going back to our tree, we wanted to know, some of these differences. So let's have a look at 370 over here, 370 versus 253, okay? So here's 370, and now it's not easy to read this thing, but I think the second column is 253, and we see there's 8. It's not easy to read this thing. So what tool do we have that works nicely with tabular data? R. Okay? So, let's fire up RStudio. And I've done all of the work, so let me show how I got to here. If I take that file from the Bactopia runs Snippy, and then the snp dist folder, okay? The core snp distance.tsv. I'll put this in the notes when I get around to updating the website. And I found a nice little, where's my code one? I found a nice little R package for plotting heat maps, okay? So, yeah, I use Pacman, like you guys saw the other day with Nazar, okay? To load the RIO. If you remember, RIO lets you import things, RIO, and then tidyverse, because tidyverse for everything, and then P heat map, okay? And so I just did, maybe I can split my code up across lines.
Don't you want to put the liter pipe at the end of the top line?
Oh, sorry, I don't know. Yeah, you know this. Is that
No,
better?
just put the enter at the other
At the other side.
Yeah,
Okay.
yeah.
Okay. So if we look, just remembering what this thing looks like on the terminal, the first column is just the name of the software, snpdist 0.8.2, okay? Which is not what I want. I wanted to rename that, so I renamed that column, but then I wanted to turn the first column into the row names. So I don't actually need this thing, but I just wanted to make a matrix with the first column as the row name. So the matrix looks like this. So immediately this is already easier to read by like loading it into, you could load it into Excel or LibreOffice Calc as well, but so we can now see, yes, 370 to 253 is 8, okay? But let's make it visual. So once I've got this matrix, I'll just run all of this code all at once. I can use pHeatMap to plot a heat map and put the numbers in place as well. And splits approximately are similar to the phylogeny, but we can see here, you know, those 253 is very similar to 373. Okay, six steps difference. If we remember the picture, where's the tree gun? Was up here somewhere there. 253 and 373. Okay, they're close together, but then 253. Sorry, 370, and then 370 is 8 from 253. Okay, so where's 370 from 373? It's 12. Okay, so what I'm saying is that 373 is close to 253. 373 is 12 from 370, okay? So it's a bit further away, but 253 is close to 370 and it's close to 373.
Yeah.
Do you now see why that 40 is there? Because 373 is definitely not close to 370, but should 253 belong with 370? Or should it belong with 373 like it is now? Okay, so that's what I mean by it's not exactly a polytomy, but it's still they're the three of these guys, they form a group. Okay, the split into pairs is not as certain as the fact. It's not so certain what the split into pairs is, but what is very certain is that they're all grouped together. Okay. So if we look at the other one, so if we look at 373 versus the reference, it's 16, but versus, why is the screen's blurry over here? Versus the 364, it's 23, and versus 260, it's 25. So these three guys, this one and this one, and we see there from the dendrogram at the top, they're all grouped together. And then these two guys are quite different to them. But if we look at these two, 260 and 364, then we see that 260 is 4 SNPs distance from 364 over there, right? The two are just 4 apart, but they're quite different to that other clade that we saw, okay? And the other thing that we see about the length is that if we look at the difference between 364 and the reference, it's 24, 260 in the reference is 25. But if we look at like this other clade over here, 370, 253, and 373, 15, 13, 16 from the reference, okay? So we see that the two clades, the different like amount of SNP difference, okay? But this is still not putting time onto the tree, but it does show us that we have two, looks like kind of two different sampling points in a way. Because there's one group that are like this and another group that's like that. And since I happen to know that these were collected at different times, that it is maybe the accumulation of mutations over time. But my point about this is just that don't stay in one world. Don't stare at text on a screen when you can use R to visualize your data for you so that you can understand it easier, you know? I mean, and if you don't want to do it in RStudio, you just switch to the right window here. Now that's Cameroon's data. Just talk about the dist. Okay, this is not, the Galaxy is not working today, which is why R was nice. Normally, I can just load my SNP distance matrix here and run the heatmap2 tool in Galaxy and do a heat map there. But today, Galaxy is not playing nicely with me, okay? But use many tools, okay?
How to link up.
How did I get the data from my data to R? I copied it.
You just copy.
I just copied it. I didn't make any fancy linkage. I just went to the output directory of the Bactopia. And remember, this is also why I was teaching you guys about SCP and things like that. Remember, you might be running this on the server. So Mary, when things were kind of running for her before things kind of broke down, was running on the server in, I think, America or something, okay? But that's why I want you to know about SCP, that you can move data between different computers, okay?
Yeah.
In this case, it was all on one computer, so I just copied from my Bactopia Vibrio directory to where I was running R, and then visualized it, you know? But my point is just learn all the tools and get them all in your toolbox, okay? On that theme, you can just close this window. I don't know why heat map is taking so long to run, because I've actually got genome assemblies running over here, and they're running much better. But anyway. Okay. On the theme of using the dates between different things, one more thing that I want to get to sorry, down here. So consensus.fa, let me just, if we take this consensus.fa, if we remember from the Snippy documentation, that is the file with the with all of the variants inserted and things like that, okay? It's gzip, okay? Which means that I cannot use more to look at it, or cat, but try and look at it with more. Then I get junk, but I can use a tool called zmore, which unzips it, and then I can look at it, okay? So here's my consensus. Unfortunately, it's kept the name of the reference there at the top, NC016445, okay? And I can also use zcat to look at it. So I wrote a little script, which I will share with you. Let me just get back to my GIST window. Just add something. Where's the NGS page? Just going to bring this back to this side so I can work on it. I want to make it easier to work with the consensus data that I've got from running this workflow. So I wrote this little script. So let me go fetch it and show you what it does. Oops. Wget like before. Oops. This is the wrong window here. I wrote this little script called gather consensi. Okay. And what I want to do here is grab everything from the Bactopia Snippy output and gather all the consensi together in one folder where I can work with them, okay? But there was one problem that I noticed that the header line was wrong. The header line has the same name as the it has the same name as the consensus sequence that I used, okay? So my script changes the header line as it reads it, okay? Which is what this sed command said. Yeah, sed is a command for doing search and replace on a string. Don't need to worry about exactly how it works at the moment. Just download this from the link that's in the Google Doc, and once it's downloaded, and if you run it, it shows you nothing, but then afterwards, there's a folder called Gather that has all your consensi waiting for you, ready to use, okay? Now, what can we do with those consensi, okay? We bring this back to my side so that I can work with it. There's a great web resource called Pathogen Watch that does analysis of various pathogenic bacteria. Okay, if this thing will load. But the thing with Pathogen Watch is that you can put fast queues into there, but it's really slow if you put fast queue data into there. It takes a long time to come up with data. And normally it isn't this slow to load either. Hello. Well, while it's doing this loading, I'm going to get my data ready. So if I go to my file browser, finally it's loaded. In my file browser, I am now going to go to, here is my Vibrio directory, and I'm going to go to this gathered folder that I've just made. And here in Pathogen Watch, I'm going to go to the upload, and I'm going to just drag my data from my file browser to this web page. Oops, that didn't do what I expected it to do. I think I must drag it onto that picture over there. No, it's still not doing the right thing. Maybe I must click here. Oh, I must click upload. Upload, upload. Now I can drag and drop. Okay, so now that I'm on this page, I can drop my data there. And it's uploading just the consensus sequences, right? And as it uploads, you can start seeing the analysis running. It already has identified that this is Vibrio cholerae. It has identified that it's Vibrio cholerae sequence type 69 using MLST. And it's busy doing some more analysis, just waiting for everything to get there. Because the pathogen watch has built has built tools for all sorts of different pathogens. Vibrio cholerae is one of them. And it can do a whole lot of like standardized analyses, and except it's saying that that's a, and it's telling you what it's busy running over here, but I'll wait for it to finish running before I look at the reports. Damn it, our network is really slow today. Just wait for it to finish the last analysis. Okay, analysis complete. Now I can view the genomes, and I can look at a report for each genome, and it can tell me what the MLST profile is there. The serogroup is 01. Cholera 01, it does a scan of some commonly known antibiotic resistance types in Vibrio cholerae, and one of the things I quite like about this is that I can do the same report on using Galaxy with, or on the command line using, say, AMR Finder Plus or something like that. But what I like about what the Pathogen Watch team has done is that they've also taken some of the single nucleotide. So when you're looking at AMR finding, there's two ways of looking at AMR finding. The one is which genes, like for instance, the VARG gene in cholera causes this resistance, but also within the GYRA gene, these particular mutations produce some partial resistance to antibiotics. Okay, so the GYRA and the PARC and on. So these are point mutations within the bacteria that are associated with generally partial or complete resistance to certain antibiotics. Okay, and most of the antibiotic resistance finding tools that work with bacteria, they only have the gene by gene resistance database for Vibrio cholerae. They've got a more complete resistance database, and then gives you a readout on the virulence genes that are identified, all of these kind of cholera-related things, and down at the end, you can run clustering based on CG-MLST, core genome MLST, to see what other samples there are in their database that are similar to your samples, okay? Some of these tools, I wish that we could run them outside of Pathogen Watch, But yeah, for now it's just Q position 500. So it's obviously a lot of people are hitting this website today. But yeah, I just wanted to illustrate that Pathogen Watch is a very useful web resource, but it works much better if you start from a consensus, from an assembled genome, than if you give it fast queues, because the fast queues, its resources, it takes for a very long time to process fast queues. Better to do your fast queue processing yourself, and then do your and then get your reports from this. Okay. There is another website called VicPred. Okay, well, I think this something on the back end is obviously overwhelmed today because it's running a lot slower than normal. Okay, I can try again, but GC content. Let's see if that works. There's another resource, a web resource for Vibrio Colorado called VicPred, but this one is, to be honest, even slower than Pathogen Watch. But they do promise me, the authors of this tool, they promise me that they will be releasing some of their tools and databases as open source in the second half of this year. Okay. What I like about this, when we do get access to this thing, it will allow us to run more command line tools to characterize Vibrio cholerae. Okay. At the moment, there's not a lot of command line tools that you can use to on Vibrio cholerae. But this one, it is, to me, it's really slow. It works on a single genome at a time basis. The mouse. And so you can only do one thing at a time. And then if you do this thing, it uploads. You click submit. And after a while, you get a report on the O type and a whole lot of more information about your sample. This is obviously Vibrio cholerae specific, whereas Pathogen Watch has been built for a number of different bacteria. So, but it is. I just do find it's useful for. Actually, one of the things I found very useful for here is just to be able to get more resources like related to Vibrio cholerae, and I'll show you how we can use that later. Okay. Yeah, it takes a little while to get an answer. Because you characterizing your genome, you want to have the databases that are species specific and all of those kind of things. Okay. So now I've gone through a lot of things quite fast here. But just before lunch, I'd like to know if you have the output of running the Snippy pipeline, please put up a blue sticky. If you don't have the output and you've had problems, please put up a yellow sticky. So I want to know who managed to actually get data out of the pipeline, because I want to use that data this afternoon. So in other words, if it ran all the way, it did all the snippies, did your gubbins, did your IQ tree, then please put up a blue sticky. Okay. So there's a problem here and a problem there. And here, what did you get? Like what?